Hello students, the topic which I am going to discuss today is about plant growth and the growth hormone, mainly auxin. Growth is one of the most important characteristic feature of all the living organisms because only the living organisms exhibit this character growth. Growth in a plant is the outcome of cell division, enlargement of new cells and then differentiation into different types of tissues according to the different types of cell wall material. This process of growth is accompanied by a permanent change in size, that is permanent increase in length or volume, and increase in dry weight of the growing parts. Then where does this growth takes place? That is the growth regions. Growth is confined only to meristems. The extreme apices of root and the shoot are occupied by primary meristems. Secondary meristems, that is cambia, give rise to the additional vascular tissues and the two protective layers of cork cells, thereby increasing girth of the trees. The activity of the meristems influence the activity of other meristems near to it, giving rise to what is called growth correlation. Now, kinetics of growth, that is the course of growth. Usually, under favorable conditions, there is a characteristic course of increase in the plant in any of the growing parts. At first, the growth is very slow, and this is called lag phase, and it takes a lot of time, which will be followed by a log phase, and here the growth increases abruptly and it will reach the decreasing of growth rate where it slows down, and lastly steady state, and here the growth is almost at a stop. When we plot the growth rate against time, we will get a S-shaped curve, which we call it as grand period of growth, otherwise known as sigmoid curve or grand period curve. The growth of the plant is controlled by various plant growth hormones or phytohormones, because it is called phytohormones as it is synthesized in the plants. According to Pincus and Thieme 1948, phytohormones can be defined as organic substances produced naturally in the higher plants, controlling the growth and the other physiological functions at a site remote from its place of production and active in small amount. These phytohormones have also been termed as growth promoting substance, growth regulators, growth factors by various workers. There are many naturally occurring plant hormones, and they are auxins, gibberellins, cytokinins, ethylene, abscisic acid, brassinosteroids, phenols, etc. And today I am going to discuss only about the auxins. The discovery of auxin. In the last quarter of 19th century, Charles Darwin studied of tropisms in coleoptile. Coleoptile is a protective cap-like which covers the plumule in the Gramineae family, that is grass, for example maize, wheat, rice, oat, etc. He performed a series of experiments. Number one, in 1880, he illuminated the coleoptile of Avena in the unilateral light, and he found the curvature of the Avena towards the source of the light. In the same year 1880, he covered the tip of the coleoptile by tin foil and induced the unilateral light. Here also he found that no curvature is found. In 1910, Boyson and Jensen also performed an experiment. He cut the Avena coleoptile tip and he placed a plate of gelatin in between the Avena coleoptile tip and the main stalk, and the unilateral light is induced, but he also found the curvature of the Avena. Then these are all under the light experiments, and in the dark, two physiologists performed the experiments. Number one, Paul 1919, he cut the Avena coleoptile tip and placed eccentrically, means placed the tip only on one side and leaving the other side free. Then in 1926-28, F.W. Wendt also performed in the same Ebina coleoptile. He cut the tip of the coleoptile and placed in the agar block and keep for some time. And after that time, a piece of the agar block is placed on the cut stump of the Ebina eccentrically. And here also curvature occurs even in the dark. From these physiologists found that some stimulus or a substance is present in the shoot tip of the coleoptile which induces growth curvatures. The chemical nature of natural auxin. In the year 1934, Kogel first of all found this crystalline auxin which he obtained from the human urine and shown to be indole 3 acetic acid, which is abbreviated as IAA. Other chemical compounds closely related with IAA are IPA, indole pyruvic acid, IBA, indole 3 butyric acid, indole 3 propionic acid. Synthetic auxins are synthetic chemicals of diverse chemical structures which behave as auxins. They are 2,4-dichlorophenoliacetic acid, abbreviated as 2,4-D, 2,4,6-trichlorobenzoic acid, abbreviated as 2,4,6-T, naphthalene acetic acid, abbreviated as NAA. The physiological effects of auxin. Auxin affects a large number of characters. The number one is stem elongation. Exogenous application of auxin causes stimulation of coleoptile and stem elongation. The most effect is obtained in stems and the coleoptiles of etiolated seedlings, that is dark grown seedlings. However, high concentration inhibits growth instead of causing stimulation, and it is believed to be associated with the hormone ethylene formation. The second effect of auxin is rooting. The auxin IAA has been identified as rooting hormone. Exogenous application of IAA induces rooting in several stem cutting. It has also induced adventitious root formation, increased number of root formation, but it inhibits root formation in intact plants. Now, the IAA is sold as rooting hormone commercially. The next effect of auxin is the apical dominance. By the presence of the apical dominance, there is suppression of the formation of lateral buds, and this is due to the presence of auxin. If the shoot tip of the plant is removed and if exogenously IAA is applied, the same suppression of lateral bud formation inhibited. Then if the cut end is removed completely, then there will be formation of lateral buds. So this is one of the example of apical dominance. The next physiological effect is about the coleoptile curvature effect. This I have discussed in the experimental works. Now the next physiological effect is in flowering. Exogenous application of auxin will induce female flower formation and increase the wall formation of the ovary, thereby leads to the formation of parthenocarpy fruits, that is the seedless fruits in cucurbits and in the grapes. This is related with the formation of ethylene hormone. The last physiological effect of auxin is in the plant growth movements, which is related with the phototropic and the geotropic. Solodny and Wendt theory in the 1927, they found that the auxin is more present on the concentrated side or in the shaded side. That's why bending of the coleoptile takes place on the shaded side and the bends towards the illuminated side. Thank you.
Thank you.